Hello, plant physiology students. This is chapter 16, Vegetative Growth and Organogenesis. And this is an overview of shoots and the terminology for shoots and leaves. And it begins, I hope you can see that, in the shoot tip, in the shoot apical meristem, which is somewhere there. You can see that you have leaf primordia that are surrounding it, embedding it, and it's actually quite hard to see the apical meristem without the use of a microscope. So the apical meristem produces leaves at the flank of it, typically in a spiral fashion. And then after that, the leaves enlarge by cell division and elongation. And the same is true for the stem portion too. So after a while, it has been growing quite a bit, as you can see here. And that's a so-called internode, the space between the leaves. And where the leaf is the node. So you have a node here, and you have a node here, and you have an internode there, you have a node here, and an internode, and a node here. Okay, and if you look at the axils of the leaves, at the base of the leaves, you have an axillary bud. Many times it's not as well developed as shown here. It can be actually just a little apical meristem or a small bud at it. And if you look at the leaf, you have a midrib shown here, and a margin out here, and let's see. And then you also have, of course, the distal proximal axis is the same as is used in animals. So the distal portion is, for example, your fingers in your body, and the proximal portion of your body would be your central body. However, in plants, you don't use dorsal ventral terminology. You use abaxial and adaxial. And adaxial looks like it refers to the upper side of a leaf, but in truth actually refers to the inner side of the leaf up when it's formed, up here in the meristem. And abaxial is the outer side of the leaf here. And it ends up being the upper side and the lower side. One way of remembering it is that the abaxial side, which is outwards, starts with a B, and if you go down in alphabetical order, the inner side will be the adaxial side with a D. Okay, and you have, of course, the apical basal polarity of a plant. You can actually turn that around too and say apical basal polarity for a shoot, and then you can say apical basal polarity for the root towards the shoot too. So you have to specify when you use this terminology what you actually mean. Okay, so most plants have simple leaves. It means that they have a single blade of a leaf, like shown here. And the blade then has a mid-vein, approximately there, and secondary veins that goes on like that. Those can be quite visible because they have not just the midrib for the mid vein, so sort of a ridge on the bottom side, but the secondary veins also tend to have ridges. But finer veins like tertiary veins and quaternary veins and veinlets, then you have to look inside the leaf to be able to see them. Then you have a petiole, the shaft of the leaf, and in some plants also a stipule. It's a small leaf-like structure at the base of some leaves. Some plants don't have it, other plants have it and are quite pronounced. For example, pea plants, pretty stipules, and the pea family, a very large family of plants, including trees, are characterized by that stipule. So if you don't know what a tree is and you see that they have a stipule, you have a fair chance that it's actually a pea relative. Okay, so on stems you can also find sometimes, especially inflorescence stems for flowers, that the leaves there don't have a petiole, and they're called sessile leaves, sometimes cauline leaves, and so they don't have any petiole. In monocots, you have a stem, at least initially, made up by the sheath of leaves. As you can see here, it's a part of a leaf that goes all the way around, and then it ends up. So that's the sheath, the bottom part of it. So it can go for quite some distance down to the meristem, and then you have a blade. Excuse my bad drawing here. And in between you have something, you can't see it here, but you tend to have something called a ligule that in some plants is present, in other plants not. And the thought is that it prevents rainwater from getting in between, so that it doesn't rot inside there. Other plants, if they don't have it, then they actually welcome any water they can get. For example, pineapple plants, they let the water lead down here, so they collect it this way. Okay, so in the monocots, leaves, you get a new leaf inside the previous leaf, so it comes out like a new sheath, and then you will get a blade expanding, and so on it goes. And eventually, though, there will be a true shoot growing all the way inside here, coming up with an inflorescence. And initially, before that happened, you actually have a shoot apical meristem way down at the base of the plant that makes this sheath and blade of the leaves. Okay, so that's monocots. They're quite special in that sense. And that's also why you can, for example, mow a lawn with grass and not kill the plant, because you're actually not removing the shoot apical meristem. You're just removing mostly the blade and some sheaths, and then new leaves will come up. Okay, there are some plants that have so-called compound leaves down here, you can see. And that means that they have not just one blade, but several blades, and those are called leaflets, small subleaves. This one with 3 leaflets, 1, 2, 3. And then it goes up into higher number, and sometimes you actually can get a leaflet like this with leaflets within that too here, so quite advanced. And I have covered the names here because there is no point remembering those different names for arrangement of compound leaves. But it's related to the mechanism of leaf formation from shoot apical meristem. It's just a repeat of that process. Now we're going to have a closer look at the organization of a shoot apex or shoot apical meristem and leaves primordia around it. So SAM here stands for shoot apical meristem, and when people talk about the RAM, they talk about the root apical meristem. The meristem itself is actually a very small structure, a little dome here. Sometimes it can be flat too, but here's dome shaped. This is a tomato plant. And at the flanks of it forms leaf primordia. Here you have the most recently formed leaf primordia, still developing, P1. And this is the second most recent form, P2 over here. And then you have P3 here, and you can see that it has so-called trichomes, hairs on the leaves that are fairly large already. They will be much smaller compared to the leaf blade later on. Okay, and you have the fourth leaf primordia, fourth most recently formed leaf primordia broken off here so that you can see the shoot tip much better then. So some people say that, yeah, you have all, or your textbooks may mention the shoot apical meristem is where all the cell division happen. And then you can perhaps believe that, yeah, there is no cell division elsewhere. It must be that the leaves grow just by cell expansion. That is not true. The shoot apical meristem do divide a lot to form cells for the new leaf primordia and for the stem, but there are cell divisions elsewhere. For example, in this primordia, a lot of cell divisions that are going on in this leaf primordia to form the leaf blade, followed usually by then cell expansion before the leaf cells fully differentiate. And here you can also see that what will become the internode. So here you have a most recently formed leaf P1. And P2 is over there, so the little space there between those will actually become the next internode, and then it goes on like that. So you will get between P2 on the other side and P3 another internode, and so on it goes. So then those internodes are formed by quite a bit of elongation between the leaves. And typically, the leaves are formed in a spiral pattern or spiral phyllotaxy, and with an angle of 137.5 degrees. Not exactly that, but if you take an average of them, you will find that it comes out to 137.5 degrees between primordia. This is most easily seen in, for example, cacti, where you don't have the leaves obscuring it and the internode obscuring the phyllotaxic pattern. Okay, let's go to the next slide. So here are some old experimental evidence that are still highly relevant, and it's based on using colchicine-induced polyploid cells. So colchicine is a drug that prevents cell division, but replication of chromosomes can still happen. And when you treat a plant with that, you can have stem cells up here, but when they duplicate the genome without cell division, they can become polyploid. And not just that, they can also produce daughter cells that will all have the same polyploid configuration. So this is a control plant, and I'm going to come back to it. It's actually layered in that you have an L1 layer, the top layer here, and an L2 layer. And the cells in those layers tends to divide anticlinally, so perpendicular to the surface. So you have a surface here, and you get a cell division. If this cell is to divide, it will produce two daughter cells in between there. And so therefore, all the daughter cells of the cells will stay in the same layer. And the same is true for the L2 layer too. So you will get cell divisions like this. Oops, that's so good. So, but still anticlinal. Whereas in the L3 layer, the third layer, it's actually not a true layer. Then you can get fairly random cell division planes, so to fill up the interior body of the plant. And most of this part here becomes the stem, but also a little bit of the leaves too. Then, so back to the polyploid cells. So if the polyploidization happens in the L1 layer, and they continue to divide, they produce offspring like this, or daughter cells, I should say. Then you will get those enlarged polyploid cells clearly visible like this. So I didn't mention that, but polyploid cells tend to be larger than diploid cells, and in this case you have an octaploid L1 layer, and you can see that the cells are much larger. Related to that, you know that wild strawberries, they're actually diploid and pretty small. The strawberries that you eat are octaploid, and that has an effect on pretty much everything in the plant. Leaves are larger, and the fruits are quite a bit larger than wild strawberries. Of course, breeding has also been used to select those that produce larger fruit for other reasons. Okay, so when you get octaploid cells in L1 layer, that is actually a good evidence to show that it is layered like that. So if they divide just anticlinal like this, cells will stay in that layer. And the same is true if you have that polyploidy happening in one of the stem cells for the L2 layer. So it has to be close to the tip here, and they will produce then daughter cells like this, and the cells will typically stay in the L2 layer. So you get that is an indication that you have a layer there too, the L2 layer. Whereas if it's if you get the polymerization in the third layer, the L3 layer, which is not really a layer, you can see that you have a cell division plane here. Might well be that we had another cell division plane there, and it could have been a cell division plane, for example, there too, and here, and so on. You see that they're not particularly organized. They're used to fill up the interior of the plant. So a question that comes out of this observation is that lineage seems to be important. So if a cell in the L1 divides, it will only give rise to L1 epidermal cells, and if a cell in the L2 layer divides, it will only give rise to L2 cells, etc. So a central question in developmental biology is whether cell fate is determined by lineage or position. And we're going to have a look at that. Here's another slide to illustrate the layered structure we shoot apical meristem. First, a drawing here, a generic one that shows that anticlinal divisions here in the L1 layer and L2 layers gives rise to two layers of the so-called tunica. The L1 layer that you can see here going into the leaf primoria, and the L2 layer here. And as I said before, the L3 layer is not really a layer. It fills much of the interior of the stem as well as a portion of the leaf and flowers interior of that. Okay, here you have a real slide, longitudinal section of a shoot apical meristem of the inflorescence of Arabidopsis. Is actually dome-shaped here, whereas the vegetative meristem that makes the leaf is actually quite flat. Okay. So here it has some false color to show that you have an L1 layer, an L2 layer, and then the L3 corpus down here. And what is not false color is what you see here, the black part here and the black part here. This is part of a so-called in situ hybridization, where a probe that is labeled radioactively is hybridizing to the mRNA of a gene of interest, and this gene is expressed for a gene that is involved in leaf primordial formation. You can see that you have expression here even before the leaf primordia has emerged. And here is a little older leaf where you also see the expression of this gene, and up here you can see a little bit of expression in the interior of this leaf primordial. Okay, so the L1 gives rise to typically to the epidermis, and the L2 to part of the interior of leaves, and the L3 to much of the stem, the interior of the stem, but also part of the interior of the leaf as well. So back to the question that I asked before, is fate determined by lineage or position in plants? You can use chimeric plants having an albino L2 layer to assess these questions. This is a fairly old experiment. So this is based on the fact that a L1 layer usually lacks chloroplast. It has plastids, proplastids, not fully differentiated plastids, but they have not differentiated chloroplasts. So that layer is uncolored. You can see it down here. It doesn't have any color, it's fairly transparent. Whereas the L2 layer has plenty of chloroplast, and it's not really shown here, but L2 layer is part here, the palisade here. The L2 layer also has giving rise to spongy mesophyll cells on the bottom side here. But when you have an chimeric plant with an albino L2 layer, so meaning that the L2 layer does not make chlorophyll or make a chloroplast, it will also be transparent or white. Okay. And then all the green color that you see in a leaf then comes from the L3 layer here. Okay, that is what is left of it here. And this is easily seen in some decorative geraniums that have been selected for this kind of chimerism, the defective L2 layer that is not photosynthetic. And that ends up being leaves with a sort of a white margin, of pale green margin here, and the interior green here. And the interior green here comes from the L3 layer, cells from the L3 layer, whereas cells from the L1 layer and L2 layers are not green here. However, over the L1 layer, you have mostly anticline cell divisions. Occasionally, at low frequency, they divide pericline, parallel to the surface of the leaf here. And then one of the daughter cells will be pushed into a position more similar to the, so they don't really move, but just by the pressure, they will end up in a position that is interior rather than at the surface. And they end up in the L2 layer. And the funny thing that happens then is that the daughter cells of that cell pushed in here will differentiate chloroplasts. So the mother cell in this layer didn't have differentiated chloroplasts, but the daughter cells will differentiate chloroplasts. Normally you wouldn't see that because all the cells in the L2 layer would be green, but in this case you have a chimera with albino L2 layer. And then you will see these daughter cells from a periclinal division going into the L2 layer as small green islands in the L2 layer. And in a uranium leaf, you can see those islands at the margin in different places here. Okay, so the conclusion of that experiment is that although the cell divisions occur typically in an anticlinal fashion and the daughter cells stay in the same layer, the cell fate is actually determined by position. So if a cell ends up in the L2 layer, it will take on the fate of the L2 cells, differentiating chloroplasts, but also, in this case, also making the palisade cell typical shape than here. So the conclusion from this fairly simple experiment, that people have looked at it in quite some detail, is that position, not lineage, appears to determine cell fate in the leaf of plants. Yeah, so this is a leaf of an L2 chimera, for example, in Benjamin ficus, bread varieties that have this decorative white margin of the leaves. And what you see in the interior here, that is the green part, are the daughter cells of the L3 layer in the meristem. And you have more or less large part of the L2 layer here. So in this area, for example, you have a fairly large sector of L2 cells here, the albinos, that's why you can see them, where they are. So this is called an invasion, L2 invasion of the L3 domain, for example, here then too. Whereas in other parts of the leaf, you can see that the L3 region makes up much of the interior of the leaf here. So it's quite variable how they contribute to that. And the rest of the slides here are a little bit misleading. It shows that the L1 layer is green, which it isn't. It's transparent. The plastids are not differentiated. You have the albino L2 layer and you have the green L3 part here. And again, here you have the L1 layer shown as green, but it is transparent. And but you can see then the contribution of L2 in this cross section of the leaf here. And the L3 provides this part here, some of the interior of the blade here, and for example also the mid vein here. Okay, so this is known as L2 invasion of the L3 domain. But you can also see in this this experiment that L2 cells, when they it's not so easy to see here, but when we take up the domain of L3 cells, we will make L3 type cells then in here too, okay, in this region. So this is another indication that cell fate is determined by position and not lineage. So the question is, how do you maintain this kind of plants? How do you propagate them to sell them as decorative plants? And since it's an L2 chimera, and the pollen cells and the egg cells may well come from epidermal cells, or they may come from just L2 cells, you will get then daughter plants that are either completely, or daughter cells, offspring that are either completely albino or completely green. So that doesn't work. So all plants that you see that looks like this, they come from vegetative propagation. means that shoot tips have been taken off and they have been rooted, put on oxygen medium to induce root formation, and then you keep reproducing like that to get clones. And that's what they are. They're clones of each other, genetically identical, including the fact that they all have a defective L2 layer, albino layer. So I'm going to go over to the genetic basis of meristem organization and talk about that. So this the shoot apical meristem is subdivided based on function. So you have a so-called central zone up here that acts as a reservoir of stem cells, and which replenishes both the peripheral zone here and the rib zone as well. So that sounds a little bit strange, that you will provide cells, this region will provide cells to that one. So what they do is that they actually divide fairly infrequently in this region, but when they divide and the Dory cells end up in the peripheral zone here, they start dividing at much higher frequency on both sides here. And actually it's not a side, this is more like a ring around the central zone. And when we divide, so as the cells end up in this region, they also start dividing at higher frequency, but in fairly random planes, so that will give rise to the rib meristem, the interior of the stem, as well as part of the leaves. So slow cell division rate here, higher at the flanks of the meristem, and then you get initiation of leaf primordia at the flanks of the meristem as well. Cell division continues in the leaf primordia and in the interior to here, but then they get a little bit further away, the cell division rate will decrease, and then cells will start to elongate or expand until they have reached their full differentiated cell fate. I'm repeating myself. I'm coming back to the layered structure just to point out that it's not always two cell layers. It can be between 1 and 7 layers, depending on the size of shoot apical meristem, and this differs in different families and species. But in many dicot plants, it consists of just 2 layers. So how do we know that you have a low frequency of cell division in the central zone and higher frequency in the peripheral zone? These are old pictures from an old textbook. It's sections. This is a longitudinal section of the shoot apical meristem. So going down from L1 layer, L2 layer, and the corpus down here. Then. And this is a section, a cross section, grazing. So it's taken over here, a cross section, and you can see this is the central zone here and the peripheral zone here. And in this experiment, before they were sectioned, the plants were fed with tritiated thymidine, so a nucleotide for DNA synthesis that is tritiated isotope, and that means when it gets incorporated, you have essentially radioactive DNA, and if you section that, you fix the cells, you embed them, section them, and apply a photographic film, typically as a thin emulsion, liquid film, and then let the isotope decay over a day or a few days, and when it decays, it will release photons, and the photons will blacken the film, and then you can develop the film on the slide, and you will see where the photons were released as black dots here. So it's not particularly convincing, perhaps, but you have a central zone here, and there's not much of incorporation here, whereas you have cells that have divided, have taken up the radiolabeled thymidine here during replication of cells in this region, a little bit in this region too, and also here. This is what is going to give rise to the midvein of this leaf primordia. Perhaps more convincing is when you look at the cross section, you see that there is not much incorporation of radioactive DNA here during replication of DNA. So not much of cell division going on here, but quite a bit in the peripheral zone here. So a lot of incorporation of radioactive nucleotides here. So interpretation of this experiment is that they're rarely dividing initial cells in the central zone. And that's what we typically call in plant science, or in stem cell, we call them initial cells. And they're frequently dividing daughter cells in the peripheral zone. So this part here, or if you look from above, central zone here with infrequently dividing cells. And when they do divide and the peripheral cells in that region gets pushed into the peripheral region, then they start dividing at higher frequency. I already talked about this on the blackboard before, but much of what we know about the genetic basis of plant development comes from a model species Arabidopsis thaliana. And Arabidopsis thaliana has very small seeds. They're somewhere between 1 and 2 millimeter long. And you can mutagenize those, with, for example, with a chemical called EMS that changes, I can't remember which base into what base now, but it causes point mutations. And in the shoot apical meristem in the seed here, it's very small, between 2 and 4 cells. And if you have a mutation, say in the genome of this cell here, it will give rise to a mutant sector later on in the inflorescence when it flowers. So you will have a sector here where all cells carry the same mutation, and you will have on the other side here a non-mutant sector. So if it's just 2 cells here, half of the plant will be mutant sector, and if you have 3 or 4 cells, you will get then a little bit smaller mutant sectors. But if you collect then seeds from individual seed pods, or siliques as they're also known as, then since the Arabidopsis is very good at self-pollinating, you will actually get segregation in the offspring, the F1 generation. So this is the parental generation, it self-fertilizes, and you get segregation in the F1 generation here. So if this is a recessive mutation, you need, let's call it 2 small a's here, and you get a phenotype here. Whereas the heterozygous plant doesn't show a phenotype. You have 2 quarters of that as well, and then you have 1 quarter wild type plants here. And the way to find out if they are heterozygous for mutation or wild type is to grow them up and let them self-pollinate again. So heterozygous plant will result in the same sort of segregation of the mutants here. So you have the red seeds here indicating the mutants. Whereas a wild type plant, of course, will only give rise to wild type offspring like this. And one thing that I mentioned in class is that if you have a mutant like this one is actually known as monopterous, and I've been working with a little bit. It lacks this part, the embryonic stem or hypocotyl below the cotyledons, and the embryonic root here. So it can't survive on soil, and that means that you can't propagate this one to produce seeds. And actually, if you keep it in a petri dishes, this is what I did some years ago, keep it in petri dishes and let it make a few rosette leaves and cut it off between the rosette leaves and the cotyledons and put it in a root induction medium, it will make a root. It has a capacity to make a root. But the inflorescence that it makes is pin-shaped. It doesn't have any flowers on it, so it's sterile. So you can't propagate like this. This is true for quite a few mutants of interest. So the way to maintain those mutants and mutations is to propagate the heterozygous plant. So you take the wild type looking plants that you have, you grow them up again, collect seeds from individual plants, and then some of them will segregate again for the mutation, or 2/3 actually of these ones, and 1/3 will segregate as wild type. So you can, what we do in Arabidopsis is usually that you surface sterilize the seeds with a little bleach and alcohol, and plate them on a sterile medium with a little sugar and vitamins and minerals. and you grow them up to seedlings like this, and when you look under the microscope and screen for mutations, and you look for what should be close to 25% of mutants if it's a recessive mutation, and then you repeat it. You take these plants, grow them up again, and see if you can recover the same phenotype in the F2 generation. Bushveld is German for bush, and it looks a little bit bushy when it grows up into a plant like this. So it's a little bit strange. It has a rosette of leaves, but then instead of making just single leaves at the flank of inflorescence like this, it makes bunches of leaves, other sort of rosettes and bunches of leaves here, and occasionally also makes flowers like this. So, and this phenotype comes from the fact that it uses up a fairly small shoot apical meristem. So it has a small shoot apical meristem from the beginning, and uses up that meristem fairly quickly, and then it forms new flanking meristems that are soon used up as well, resulting in the bushy phenotype. So the phenotype looks a little bit opposite to what you expect from it, actually, that it has a meristem that is quickly used up because it makes a plant with extra leaves. Another mutant of interest are actually three genes that are mutated: clavata 1, 2, and 3, and they have enlarged shoot apical meristem. You can see that down here. If this is from the apex of an inflorescence of a wild-type inflorescence, and you have the apical meristem down here. In the clavata mutants, that apical meristem is much enlarged and is typically flattened like this. So it looks like if you look from the side, it looks like you have parallel stems going together towards this large apical meristem. What you see on the flanks here are the flower primordia. And also going to mention a couple of mutants that are not shown here. Cup-shaped cotyledons 1 and 2. Acronym for that mutant is CUC. And it lacks shoot apical meristems and have fused cotyledons, which is not shown here. And also asymmetric leaves 1, AS1, have asymmetric leaves, as the name indicates. And again, the phenotype does not really fully reveal the function of the gene. And for this mutants here, that's in part because the genes have overlapping function. So that's cup-shaped cotyledon 1 and 2 have overlapping functions. So if you knock out of one of them, you don't get the full effect of losing that function. The same is true for asymmetric leaves 1 and 2. If you knock out one of them, you only get a partial phenotype. Whereas if you do double mutants, you get a stronger phenotype. So the evidence in support of the model that I'm going to come to is that the molecular identity of the encoded proteins of these genes are typically transcription factors, just like in what you find in Drosophila and in C. elegans too. So these are genes that regulate the expression of other genes and act as key regulatory genes or master genes and regulating quite a few different or downstream genes. STM and its close related genes NAT1 and 2 belongs to a family of homobox genes that encode so-called homodomain transcription factors. And you know that from Drosophila, such as the Antennapedia mutant that has legs instead of antenna. And the Wuschel gene is also a homobox genes but belong to a different family. Clavata 1 and 2 are membrane-bound receptor kinases, also commonly involved in important steps in developmental biology. And clavata 3 turns out to be a peptide ligand to clavata 1 and 2. AS1 is a so-called MIB-type transcription factor. It's not worth remembering the MIB-type here, but it's another type of transcription factor. I'm going to go into 3 parallel mechanisms in the organization of the shoot apical meristem. First, maintenance of the initial cells or stem cells in the center of the shoot apical meristem. I already talked about that a little bit. Then secondly, the regulation of proliferation at the shoot apical meristem. And this should have been a 3 here. Specification of leaf and flower primordia at the flanks of the meristem. This slide shows a drawing of the expression domains of the clavata 1, 3, and Bushel genes. So this is the reproductive meristem, the inflorescence meristem, Arctopsis, dome-shaped like this, has made a primordia here. It looks like a leaf primordia, but will develop into a flower later on. So it's not so easy to see that from real sections here. I summarized them in just one here. Clavata 3 is expressed in the upper part here, the L1 and L2 layer. Clavata 1 is a pink expression in a fairly large region here, whereas and Bushel expressed in the red gene, a very small region down here. And here's another experimental evidence in support of the model that I'm going to come to. So this is what I talked about before. Bushel is expressed in a small region in the L3 region here in wild-type shoot apical meristems. This is from in situ hybridization to the mRNA from the Bushel gene. But if you look in a clavata mutant such as clavata 3, you can also look in clavata 1 and 2, and it will look much the same. You can see that the domain of the Bushel expression is much expanded here. So it used to be just a small region up here, and now it's expanded sideways like this. And I should change here. I said that this is the L3 layer, but it's clearly the L2 layer because you have a pretty clear layered structure here. Okay, so how does a model work, or what does it suggest? It suggests that Bushel expressed in the lower parts of the central domain here activates clavata 3 that is expressed above here in the stipe region, and clavata 3 in turn, which is the ligand of clavata 1 and clavata 2. Clavata 2 is not shown here. It will be secreted by its cells, and cells close to it here will be activated by that, and what they do in turn is that they send out a signal that at the time of this figure was unknown what it was, now it's known, inhibits the expression of Bush. And you can see that this is a so-called feedback loop. Bushel activates clavata. Bushel is a transcription factor that's secreted by cells here. It can move to neighboring cells, activates clavata here, clavata 3, I should say. And clavata 3 activates, when it's secreted, clavata 1. So those are membrane-bound receptor kinases, and clavata 3 is a ligand to that. So binds to the surface of all cells, activate clavata 1, and that leads to interaction here, suppression of Bushel. And the result of this feedback loop is that if Bushel expression for some reason expands, it will send a stronger, so this region here with Bushel expression expands, it will secrete more Bushel protein, will activate more cells, the clavata 3 expression in more cells, which in turn will be secreted to other cells, the clavata 1, activating clavata in a larger region, and then you get a suppression of Bushel. So stronger regulation here results to suppression of Bushel. So this way the domain of Bushel expression is kept in check. It will remain small. So if it enlarges, it will be inhibited this way, and if it shrinks, it will send out a smaller signal, and therefore you will be able to enlarge it. So you have a feedback control of the size of the Bushel expression here. And Wuschel is central to maintaining this central stem cell or initial cell identity in here. In the context of the mutant phenotypes, then, which is the basis of this model, then, if you think about clavata mutants, they have an enlarged shoot apical meristem, clavata 3 as well as clavata 1, clavata 2 that is not shown here. And that is because in the absence of these gene functions, you don't get the feedback loop suppressing the expression domain of Wuschel. And Wuschel then can enlarge, as we saw in in situ hybridization in a clavata mutant, get much larger. The central zone of the meristem gets much larger. And as a consequence, you also get a much larger peripheral zone. And the peripheral zone will give rise to more flowers and more leaves. And if you think of a Wuschel mutant, Wuschel is what specifies the identity of a central zone. It is expressed when the meristem is well, it's not expressed, but initially you have a small central zone. But since Wuschel is not expressed to maintain that one, the plant will use up its central zone, and then it will not produce any stem cells or initial cells for a peripheral zone, and the whole meristem will essentially get used up after a while. And when it's used up, the plant will try to make new meristem, manage to make new meristem elsewhere, initiating new leaves, but then that meristem gets used up as well, and so it goes on, leading to the Wuschel or bush-like phenotype of the mutant. Here's a textbook version of that model, and I already mentioned it twice, but please read the text here and see if it makes sense to you. So what is the function of the STM or shoot meristem less gene? And the gene actually has the opposite function of its phenotype, because phenotype shows what is lacking when the gene is dysfunctional or not functional at all, and showing then what the gene normally does. So STM is expressed from the early heart stage embryo in between the cotyledons in this region, and what it does, it defines the region for the shoot apical meristem. And its function only kicks in after seed germination, and its expression, this is in the inflorescence, but it has a much larger regional expression than also in the vegetative meristem. And it overlaps with the central zone here and the mechanism that I've talked about before then. So that mechanism still has its function, even though it overlaps with the STM expression, to maintain a central zone here with slowly dividing cells. And but when those cells leave, the daughter cells get sort of pushed out of the central zone. They end up in the peripheral zone in this in the meristem here, and then STM function kicks in, and it results in high frequency of cell division in these cells, but also to maintain them as undifferentiated meristem cells. So increases the rate of cell division and maintains them in an undifferentiated shape. And it's only when STM expression is suppressed at the flanks here where leaf primordia or flower primordia can be formed. So I mentioned that STM suppresses leaf formation, and here I'm going to show that part of the evidence in support of that. So these are RNA in situ hybridizations, so probes that have antisense sequence of mRNAs of genes of interest, and in this case they're not radioactively labeled. They're labeled with a UTP that is has a so-called dig molecule attached to it. In the dig molecule, there are antibodies against that, and then those antibodies can have an enzyme attached to it that can result in a color reaction, and you can get purple to pink color depending on how much of expression that you have. So pink here, fairly low levels of expression, purple, fairly high level of expression, and it can also be bluish. Anyway, so if you look at this case is asymmetric leaves 1, but we're looking at in situ hybridization, we're looking in wild type embryos and STM mutant embryos too. So if you look at wild type embryos, you can see that AS1 is expressed in the cotyledon primordia here, 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 and here. And it's not expressed in between the cotyledons where we shoot apical meristem. It is. See that here as well. Whereas if you look in the STM mutant, in the absence of STM gene function, you can see that AS1 expressed is also in that region here, in between the cotyledons. So a conclusion from this experiment is that STM normally suppresses the expression of AS1 in that shoot apical meristem region. Since in the absence of STM function, you get AS1 expression also in that region. So here's the expanded model for regulation of leaf primordia formation. And it's actually not the whole picture either. And I'll come back to that. So this model states that STM is expressed in the meristem. So you have STM expression in the meristem region. And its function there is to maintain a high rate of cell division and keep them in undifferentiated state. AS1 is inhibited by STM, so in this region you will not have AS1 expression. You see this inhibition here. STM suppresses the expression of AS1 here. Now AS1 in turn suppresses the expression of the NAT1 and NAT2. These are two genes that are very similar to STM. And so in the absence of AS1 expression, you have a high level of NAT1 and NAT2 expression as well. So together, STM, NAT1, and NAT2 maintains this shoot apical meristem region, the peripheral zone of it, high frequency of cell division, and also suppressing the differentiation of cells. However, at the flanks of this region, flanks of the flanks, you get the formation of leaf primordia by downregulation of STM, and that results in upregulation of AS1 since STM suppresses AS1, as you see here. So STM, when STM expression goes down, AS1 goes up, and that triggers then leaf formation. And what causes this downregulation of STM? Well, I have alluded to it before when I talked about auxin maxima formation. So what you get here is an auxin maxima formation, and that results in downregulation of STM and an upregulation of AS1, and you get then a leaf primordia to form. So here you actually can see a leaf primordium that is forming, and here you have an older leaf primordium. AS1 is suppressing NAT1 and 2, and also promoting development of leaves, or in the reproductive meristem, the formation of flowers. There are also domains in the meristem based on hormone distribution. For example, cytokinin, the hormone that is important for cell division as well as the formation of shoot apical meristem, if you want to induce shoot apical meristems. And cytokinin is high in the region where you have STM expression, and that's because actually STM activates, or it leads to the formation of cytokinin, or biosynthesis of cytokinin in that region. Gibberellic acid is high in the leaf primordia, in this region here, in part of it here. And this is actually not leaf primordia, these are flower primordia, but it's the same there too. So this is an early flower primordia, this is a later flower primordia. It doesn't really look like a leaf anymore. It's because it's forming the integuments of the flowers. So this center part will lead to the formation of the pistil and stamens, and this part will lead to the formation of sepals and petals. Anyway, back to the hormones then. Auxin distribution is a little bit different. It's confined primarily to the surface of the leaf primordia and the meristem, fairly high in the center here. and then also high where the leaf primordia forms. So that's the auxin maxima that I mentioned before, and high also where the internal vascular tissue will form. This is what will lead to the midvein of the future leaf forming, or flower, I should say, forming here. So together these hormones then influence cell division and cell fate, and this also shows a map of cell cycle rate. And so low in the center where you have the central zone here, higher at the flanks of it, we have STM expression, but even higher actually where you have the leaf or flower formation occurring then. And that is needed to result or give rise to those organs. So molecular studies then have shown that STM, part of its role is to activate genes that lead to cytokinin biosynthesis, but at the same time inhibit genes that lead to GA biosynthesis. And so by inhibiting GA biosynthesis, it also prevents GA's inhibition of shoot apical meristem identity in this region. And at the same time, STM activation of cytokinin biosynthesis lead to maintenance of the shoot apical meristem identity, the high frequency of cell divisions, as well as maintaining the cells in an undifferentiated state. That cytokinin also activates GA2 oxidase at the flanks here. This is the base of the leaf primordia. And GA2 oxidase is an enzyme that breaks down GA, so it inhibits GA signaling. And outside of that domain, this is the leaf primordia, then the cytokine doesn't influence the leaf primordia here, and GA is produced at fairly high levels. And one of the functions of GA here is to cause cell differentiation. So not just elongation, but cell differentiation eventually, enlargement of the cells. So back to STM. STM regulation specifies the meristem region of proliferating cells. Downregulation of STM at the flanks of the meristem results in a ring-shaped region competent to make leaf or flower primordia. So it's a ring-shaped region. We only looked at it in longitudinal sections, but if you think about it, it's a ring-shaped region, but it's then competent to make leaf or leaf primordia. So what specifies where in the ring those primordia will form? And that's where auxin regulation of leaf and flower formation comes into play. And I already mentioned a little bit about that. These are some slides from my lab, from an old paper of ours, that shows the expression of PIN1, if you remember that one. PIN1 is an auxin efflux carrier, so moves auxin out of cells. And DR5 is a reporter gene, a promoter that is activated by auxin and leads to expression of the GUS gene. And the GUS gene can convert uncolored substrate into a blue product. And this both pictures shows the vegetative meristem from above. It's hard to work with the vegetative meristem. Most people work with reproductive meristem epidoxis because it's larger and easier to look at. But we're foolish enough to look at the vegetative meristem. And here is the central zone from above. And then you have central zone inside here. You have a peripheral zone which should have high levels of STM expression. But then you have different leaf primordia. So here's an incipient leaf primordia, has not yet formed a leaf primordia. And you can see that PIN1 is expressed in those cells. And then you have first leaf primordia here, and second one here, third one here, and fourth one here. And these are optical sections that actually doesn't show the whole leaf primordia. So shows the internal midvein of that leaf primordia. You have a high level of expression of pin1 here, a little bit of the flanks too. And the same is true here. This is the midvein. And here's this also midvein region of this one here. Okay. And at the same time, you can see that there's also seems to be high level of auxin based on expression of the DR5 gas reporter in the incipient leaf primordia and the real primordia here as well. So what this pictures and other pictures, we have better pictures actually in the longitudinal section which shows this better, but polar auxin transport establishes auxin maxima here. So you have accumulation of auxin, and that is in part due to the localization of the pin protein directing the flow of auxin to the tip of leaf primordia, and then it gets internalized and you get auxin flowing downwards in the midvein towards the root vein. So. This picture is from a publication by Chris Kuhlmeier and co-workers some time ago. They worked with tomato. And tomato produces fairly large shoot apical meristem compared to our drops. This is easier to work with. If you grow tomato plants on a medium with that has so-called auxin efflux inhibitors, so inhibitors that inhibit the activity of efflux carrier like PIN1 and so on, you inhibit polar auxin transport. And you also block auxin maxima formation then at the flanks of the meristem. So this is a shoot apical meristem up here. Normally you would see a leaf primordia forming at the flanks of the meristem like this, but if you grow these plants in the presence of such inhibitors, the PIN1 protein and other PIN proteins don't get localized to specific parts of the cell, and the auxin maxima cannot be established, and therefore they don't form leaves if it's a vegetative meristems or flowers if it's a reproductive meristem. But when you have these pin-like structures like this, and you artificially apply auxin, has been applied here then you can get some interesting results. So this is the same picture as before. You have applied auxin here, and if you come back a little bit later, this is for application point, a leaf has formed, and it has sort of bent over the dome-like structure. So this is exactly the same meristem, just taking a picture just taken a little bit later here, and has formed a leaf here, or leaf primordium, with a lot of trichomes on the surface. And if you do this in a reproductive meristem, it looks perhaps somewhat obscene here. So you have a pin-shaped inflorescence here, and you apply auxin, and in that region then not a leaf but a flower primordia will form here. So the conclusion for this kind of experiments is that an auxin maximum is required and sufficient to trigger leaf and flower formation at the flanks of the shoot apical meristem. Here's an old picture of mine showing the PIN1 mutant. So it has made, it makes a root, makes a hypocotyl embryonic stem, and when it germinates, it will start to make, in addition to small cotyledons at the base here, rosette leaves here. And when it bolts to make the inflorescence stem, actually doesn't form any flowers at the flank of it. There's a little bit of a contradiction here. If PIN1 has a role in leaf formation and flower formation, why do we then have leaves here? But it's not the only protein that is important for leaf formation. Only efflux carriers are important for leaf formation, but it has a fairly central role in the formation of flowers in the inflorescence stem. And I mentioned root monopterus before, which is a transcription factor in auxin signaling, a so-called auxin response factor, which actually shows a similar phenotype. And that sounds perhaps a little bit strange. It's a transcription factor rather than an auxin efflux carrier. But we and later others have shown that the monopterus gene is necessary for full expression of the pin protein. So if you don't have pin protein fully expressed, you will end up in the monopolist mutant. You will end up with a similar phenotype here. There's as I mentioned before, a little bit of a contradiction. In both mutants are able to form leaves, but they don't form the flowers, and it should have become meristem. The lower part here is just an expansion of that here. So you have, this is a reproductive meristem longitudinal section, but you have, it's hard to see here, but you have a flow of auxin based on the localization of the proteins here up to the tip of the primordia, and then internally down here that will form one of the vascular bundles of the stem. You also have the expression of pin1 here too in the incipient leaf primordia, and here is a primordia that's a little bit more advanced. You have expression in the surface here, and then internally like this going down. This is a different plane of section, a little bit. Actually, this is I think this is a reproductive meristem, and this is the vegetative meristem, a little bit flatter. These are leaf primordia. And you get sort of cross section. So here you have a young leaf primordia, you have a pin1 expression fairly strong at the tip of the leaf primordia here, and these are leaf primordia that have been cut off. It's coming from the other side, and you have expression at the flanks of the leaf here. You can ignore this one. This is hard to understand if you haven't been working with this kind of structure. So I mentioned that based on pin1 protein localization, subset localization, auxin, and also DR5 expression, showing that the tip of leaf primordia has a lot of auxin in it. That auxin moves in the epidermal cells to the tip, and then accumulates there, triggers then formation of the leaf primordia in the first place, but also then when it gets internalized, the formation of the midvein. Now, when that happens, auxin is directed into this primordia. It drains the surrounding region of auxin, and so no auxin maximum can be established close to that existing leaf primordia, except when that drainage is no longer efficient, so to say. It's far away from the existing leaf primordia, such that a new maximum can be generated by flow of auxin towards a specific region here. So you get another auxin maximum formed here, and in that zone that is competent, the ring-shaped zone that is competent to make leaf primordia. So you get an auxin maxima here, and that triggers then the formation of the next leaf down here. So this is a little bit more advanced. You have an older leaf here. You have a most recently formed leaf here, and next leaf will form in a region in this peripheral zone that is as far away as possible from existing primordia here. And that is in part generated by cell divisions too. So you get a larger physical distance between this one and this primordia here, and that auxin maxima formed here, as well as the formation of a leaf primordia here. And you can already now perhaps see a part of the basis when why leaves are formed at a certain distance, a maximal distance. If this is the apex here, you can see that there's approximately, well, you can't really see it, but you will have about 137.5 degrees angle between this primordia, and then the same from here to here as well, then 137 degrees angle. Okay. You have a previous paradigm until the evidence comes up that it doesn't support that paradigm or that model or hypothesis, and then a new paradigm or model or hypothesis develops. And when it comes to the spacing of leaves and flowers at the shoot apical meristem, the previous paradigm was that many textbooks and many people will still claim that this is the case, that existing leaf primordia produce a diffusible inhibitor, preventing leaf formation in the vicinity. So here is if you start with most recently formed leaf primordia here, when it forms, it secretes an inhibitor that inhibits leaf formation in its vicinity. And that's true for the second oldest leaf primordia here too, produces an inhibitor in a field around it. But that field is a little bit smaller as it ages. And the field in the third most recent leaf primordia is even smaller than. And the fourth leaf primordia doesn't have any field like that at all, as well as the fifth primordia here. But this is still a fairly small region then. So next leaf primordia can only form in a region where you have that is not affected by that inhibitor, that unknown inhibitor. And the next, well, not P5, the incipient leaf primordia 1. So the next leaf primordia will form in the largest gap, or the first real gap formed in the field of its inhibitors. So the next one will form here, and when it forms, it will start to produce an inhibitor that inhibits leaf primordia formation in its vicinity, and so on it goes. Incipient 2, incipient leaf primordia 3 here. And leaf primordia don't form in the central zone because that's where Bushel and other genes are expressed. It maintains the central zone. And in the peripheral zone, which is somewhere here then, STM maintains high frequency of cell division, but also in undifferentiated state then. So it's only at the flanks of that peripheral zone that leaf primordia can then form. But anyway, so if you look at it, the old model is that emerging leaf primordia produce a field of a diffusible inhibitor that prevents leaf formation in its vicinity. The new paradigm, developed in large part by Chris Kuhlemeyer and his co-workers, but also others, and a bit supported by our publications as well, is that a polar auxin transport mechanism generates auxin maxima, and high concentration of auxin triggers leaf formation. So yeah, and I mentioned it before that auxin transport drains the vicinity of auxin, preventing then leaf formation. So the new paradigm is that it's the removal of an activator and not the production of an inhibitor that suppresses leaf formation close to existing leaf primordia. So if you look at the leaf primordia here, that existing leaf primordia will remove in its vicinity auxin, because auxin flows towards the tip of the leaf primordia and then gets internalized and away then. So you have a drainage of auxin around that leaf primordia. And only when you have a gap in that drainage of a inductive auxin, like here, auxin flows up here and you can then form a new auxin maxima in that region. Then you will have a formation of a new leaf or a new flower then. So if you look at the previous paradigm here, is that you have a field of inhibitor produced by existing leaf primordia. What happens instead is that leaf primordia, if you look at this most recently formed leaf primordia, drains this region here by epidermal flow or flow of auxin in the epidermis towards the tip of that leaf primordia, and then it's drained away internally. And the same is true for this ones too, although you get less of that when the leaf primordia gets older. And it's only when you have a gap in the drainage of auxin that a new auxin maxima can form. So auxin will flow and form a new auxin maximum here that will trigger the formation of a new leaf primordia.